I know like there's like 12 of you who like to take the practice exam over the weekend. So I really apologize to those of you who were really planning on taking it over the weekend and then couldn't. But yeah, it's up now. If you find any like problems with it, please let me know. The answers should shuffle. I know some of you have had issues with that, not shuffling. So let me know if you have any issues with the answers not shuffling or if there's anything that isn't clear. I'll also have like my office hours tomorrow at 5 and then Wednesday morning as well. So we can discuss like the practice exam there too, or through email, whatever you prefer. Okay, I think that's all I wanted to talk about. Does anyone have any questions before we start the last 2 lectures? All right, if not, I guess we'll go ahead and start. Okay. So with him, go through the learning objectives because those are the high yield points that he really wants you to know. So he's really good on objectives. So look at those. All right, so we've talked about the cephalic and the gastric phases of digestion. So now we're moving on to talking about the intestinal phases of digestion. So these, the entry of chyme, I think it's pronounced, into the duodenum is going to be based off of the size. So a larger size is going to increase emptying, and then something that has a lot of macronutrients is going to like be slower to entry because you want to spend more time digesting it if it's more nutrition dense. But you should know that these are the 4 things that will trigger movement of chyme out from the stomach and into the duodenum. So distension is going to trigger this. Obviously, if you're moving things in your stomach, that's going to tell your stomach that you have food in it, and so it's going to want to empty it. Low pH, so a low pH will then cause you to move things out to the duodenum as well. And then osmolarity, remember your duodenum is going to be responsible for either pushing fluid out into the duodenum if it's a hypertonic solution or absorbing water and moving sodium into the duodenal thing to make it more isotonic again. And then digestive products. So anything like proteases, lipases, they can also trigger this response as well. So memorize those 4 things. As for the responses, it's kind of like a generalized thing. So you should know about how the duodenum does deal with changing the osmolarity to more isotonic osmolarity because you need to make sure that you have sufficient sodium because you need sodium for absorption because it works as something that helps with co-transportation of things like amino acids and stuff like that to help absorb things. So you need to make sure you have an isotonic solution. Another thing is it's going to be responsible for triggering bicarb release from the pancreas to help make it not as acidic so it can move through the intestines without damaging them. So obviously that's going to trigger enzyme release as well. And then you also release bile from the gallbladder. And then it also can help with regulating the patterns of motility. So either segmentation, which is the first thing that occurs to help mix things and digest them better. And then peristalsis, which is the movement of like the undigested material down towards the colon. So what he has underlined, make sure you memorize those. And then the responses he gets into in more detail. So that's just kind of like a generalized list for now. Okay, so this is just kind of telling you what happens in the stomach. So when the food enters the stomach, it's just known as a bolus of food. So it's like partly digested from like the enzymes in your saliva, but it's like only a little bit. But once it hits the stomach, it'll be broken down even further into smaller particles. So that's just all that's saying. Your triglycerides can be broken down as well in the stomach. But your stomach won't completely break them down. So you will see some triglycerides leaving the stomach and entering the duodenum. But you will also see free fatty acids and monoglycerides as well. He also mentioned something about PUFA, but don't worry about that. That's just his interest. A lot of the stuff that he says are just things that he's like interested in and wants to like, I don't know, get you guys interested in them, but he's not going to test on that sort of thing. Okay, so with protein, also same kind of thing. You will see some peptides, some amino acids that are digested in the stomach, but for the most part, you're not going to see a whole lot of protein digestion. And then starch. You will see some starch digestion as well, but into oligosaccharides, but that's like about it. So just kind of just generalize the fact that the stomach doesn't do a whole lot of digesting, but it does get things started, so to speak. Okay, so I would say that this is a high yield slide. So we already mentioned that gastric distension is going to increase the emptying of the stomach into the duodenum. And we already mentioned that, a bigger meal is going to cause increased emptying, which makes sense because if you're eating a big meal, you want to make more room in your stomach. So you're going to start dumping it into the duodenum to make more room. But you should have an idea as to what inhibits or slows gastric emptying. So duodenal distension, right? Your duodenum can only hold so much. So once it starts to stretch, it's going to want to inhibit gastric emptying because it's starting to get full. Fat, proteins, and cations can also inhibit it because that's also kind of related to this idea here that once you get food in there, it's going to want to do sort of like a negative feedback thing where it just wants to start slowing down gastric emptying because your duodenum is starting to get a little bit more full. Acidity is also related to this. Acidity tells you that you're dumping nutrients from your stomach into your duodenum. And so that will also slow it down because remember your duodenum is going to want to secrete bicarb to help reduce the acidity. So you don't want a whole lot of acidity being dumped into the duodenum because it needs the correct amount of time to dump bicarb into that and reduce the acidity so you don't damage your intestines. Another thing that your duodenum does is it, takes hyper or hypoosmotic chyme and then corrects it to make it isotonic. So if something is hyper or hypoosmotic, that's going to also inhibit gastric emptying. Also for the same reason as the acidity is because it wants to give the duodenum enough time to help correct this, make it more isotonic, because your duodenum will need to do some extra work to get it to an isotonic solution. So yeah. And then he also mentioned that solids are more inhibitory than liquids, which makes sense because liquids are really easy to absorb and solids take a little bit more time to digest and absorb. So liquids would be less inhibitory because they can move through you pretty quickly. Solids do need a little bit more time. You should also be familiar with the hormones that are responsible for slowing down gastric emptying as well. CCK is a big one. This is something that is super important. So make sure you have a list of all the stuff that CCK does because it does a lot. VIP is also something we are somewhat familiar with. It's usually linked with nitric oxide and usually does vaso or dilation of your sphincters. So that's another thing. Gastric inhibitory peptide. This is the one associated with insulin release. So it could also inhibit gastric emptying because remember, if you have a lot of glucose entering your duodenum, you're going to want to secrete a lot of insulin, and you need enough time to do that. So it's going to inhibit gastric emptying. Somatostatin and secretin will also influence this as well. So make sure you memorize these as well because this is, I would say, is a pretty high yield slide. Okay, so this is another high yield slide. He talks about CCK and secretin a lot, both in this lecture and the next lecture. So make sure you know what these two do in terms of, the pancreas, what they do to the pancreas. So eye cells are going to secrete CCK. So you should know the cells that secrete CCK, and you should also know the cells that secrete secretin. So this one's really easy to remember because S and S. I don't have a mnemonic for I and C, but just, I don't know, make a list, I guess. So these CCK or your eye cells are going to sense fat and protein and then changes in the osmolarity of the food that's entering. You don't have to memorize these in particular, but you should know that food is going to trigger this. And it's usually fat and protein that does this because that's kind of like what your duodenum senses for the most part. So you can kind of generalize that in your head. So CCK plays a role in both secreting bicarbonate and water, and it also plays a role in enzyme and proenzyme secretion from your pancreas. So this is your exocrine pancreas, not your endocrine pancreas. So I think there was a question on the exam where he put like endocrine versus exocrine, and like it was kind of the same answer choice, but he switched it. So don't let that trick you. Make sure you know like the difference between the two and like read the answers carefully. Anyways, CCK can kind of do both. So it helps both with enzymes and bicarb, but the major one that it's involved with is the enzyme secretion. Secretin is going to be mostly involved with water and bicarb secretion. So that's like the major one that it's connected to. And then he also mentioned about how CCK can help with satiety in your brain, but that's like new-ish research. He's not going to test you on that, so don't worry about the details there.
Hey, Phoebe,
Yeah.
could you explain again what you meant about that exocrine endocrine point you made?
Yeah, so because we do talk about insulin in these lectures, if he asks you a question and makes one of the answer choices like the endocrine pancreas and makes another answer choice the exocrine pancreas, like make sure you know like what he's asking, whether he's referring to the endocrine or exocrine, because that could like trick you into picking the wrong one if you're not reading it carefully.
Okay, that makes sense. Thank you.
You're welcome. Okay. So pretty much the big point here is that glucose is going to be absorbed really quickly. So gastric emptying is going to be dependent on the nutrients of the food that you just consumed. So you can expect glucose to be emptied pretty quickly, and then things that are harder to break down, such as protein and fat, they're going to generally be slower in emptying into your duodenum from your stomach to your duodenum. Yeah, and then like once again, he mentions how like osmolarity of the chyme can determine whether like how fast you empty it into your duodenum. But yeah, if it's not isotonic, it's not going to be emptied as quickly. I think Gatorade is mostly isotonic, so it would be like emptied into your duodenum pretty quickly. So he just uses that as an example. And then glucose gets absorbed really quickly. So that's another thing there. He also mentioned this, but I would say this is pretty low yield as well. It's just like he's kind of mentioning like what this could potentially be important for in terms of like research. So don't worry about that too much. Just kind of generalize these concepts and you should be fine. Okay, so once again, your duodenum is super important for changing the osmolarity of the meal. So its major thing that it does, or one of the major things that it does, is it takes either a hypotonic meal or a hypertonic meal and turns it into an isotonic chyme thing. So that either includes, pumping water into the duodenum to dilute a hypertonic meal, or pumping like solutes into the lumen to make it more hypertonic, or absorbing water, that sort of thing, to help, make it back into an isotonic solution. So by the time it enters the jejunum, it will be isotonic. And once again, this is important because remember, sodium is going to do a lot of like work with like co-transporters to help with absorption of different nutrients, as you'll learn in the next coming slides. So that's something that the duodenum does to prepare for that eventuality and make sure that you don't have not enough sodium, or to make sure that you don't have too much sodium, so that process can be that can function properly. Okay, so you should know the difference between segmenting movements and peristalsis. That's super high yield. He like spent quite a bit of time on it, so I think it's super important. And he also underlined it, which also means that it's super important. So your intestinal motility, you should know that your enteric nervous system is involved here. And so it's going to include reflexes. So reflexes don't quite go to your brain. I think they only go to your like spinal cord. And then hormones can help influence this, and then neurotransmitters as well. So it does kind of partly connect to your brain. The rest is reflexes and hormones that can help influence how your enteric nervous system works. So the enteric nervous system is going to be connected to your circular and longitudinal muscles. So your circular muscle is going to contract, and it's going to squeeze the intestinal wall so that you are kind of like mixing the stuff inside of there. And this is known as segmentation. So associate segmentation with mixing and circular muscles, and you should be fine. Peristalsis is going to be the thing that happens after you do most of your absorption. It's mostly going to be involved with moving the stuff that doesn't get absorbed or digested along towards the colon. And this is going to be done by longitudinal muscle contraction. So associate longitudinal muscles with peristalsis, and it's the second step. So the first thing you do is segmentation. The second step is peristalsis. So don't worry about this picture. I'm sure you guys know this from histo, but it's just showing you where the myenteric plexus is located. But don't worry about it because he's not going to ask you any like anatomy questions. So yeah, your myenteric plexus has parasympathetic tone, obviously, because your GI tract is stimulated by parasympathetic nervous system. I think that was the only takeaway here. That it's connected to both longitudinal and circular muscle, depending on what you want to do at that point in time. Okay, so here he's just showing you how these muscles work. So with circular muscles, it squeezes it, so it contracts and like squeezes the lumen or the tube, and that helps move things forward and backwards and helps with the mixing process. Whereas longitudinal muscle, it's long ways, so when it contracts, it just helps move things in one direction. So that's all this is saying. It's really just him re-emphasizing this point. Okay, and once again, he shows you the differences between these two. He also mentions that gap junctions are involved to help like stimulate the muscle in a coordinated fashion, because you want to make sure that the muscle is contracting in a coordinated way so that it's not like, not doing the right thing, especially with peristalsis. If you want things to move in one direction, you need to make sure that it's contracting in a way that's like coordinated, so that one part contracts, then the next part, and then the next part. And that's facilitated through gap junctions, which move the electrical signal across cells. Okay, so the electrical signal is going to be made in the interstitial cells of Cajal. So you should definitely know these. These are part of your enteric nervous system. They are the ones that make the action potentials that are then connected to smooth muscle, either your circular muscle or your longitudinal muscle. And when they, are activated to signal the electrical signal, that will then stimulate either of these muscles, depending on which ones they're connected to. And they're like the pacemakers. So kind of in the same way that the pacemaker, you have a pacemaker region in your heart, you also have pacemakers in your gut, which are coordinating the firing of these action potentials so that you have coordinated muscle contraction, just like exactly like how you have it in your heart. So don't worry about like the calcium current here. It's just saying, we already learned this when we learned about muscle, so don't worry too much about that. But do know that these interstitial cells of Cajal are making these slow waves, or they're also known as BERs. So you can use either term to describe the slow waves that they make. But yeah, I think that's all that this is saying here. Okay, so once again, slow waves are the same thing as BER, or your basal electrical rhythm. So your interstitial cells are going to be partially activated when you are at rest. So they will fire like 1 or 2 action potentials, which does lead to a small contraction, but you can see that like muscle tone is like fairly low in comparison to when they're stimulated. So when you're eating and you want to start mixing things in your intestines and moving things along, that's when you'll see more action potentials. They'll be stimulated to fire, and that's going to lead to more contraction so that they're actually working, moving things along. However, when you're fasting for long periods of time, they're going to be inhibited because you're not going to have to move anything because there's probably nothing in your stomach or your intestines for that matter. So they're not going to have any action potential firing. You're not going to see a contraction. So pretty much the takeaway is ICCs, or your interstitial cells of Cajal, are going to generate slow waves. These slow waves, also known as basal electrical rhythm, are going to be responsible for contracting the muscle. That is, either your longitudinal or circular muscle. Okay, so this is something we've already seen before. It's the exact same one that we saw in the stomach. Like I said, memorize it again. I'm not going to go through it again because it's literally the exact same thing, but do make sure you know it. It's the same thing for all regions of the GI tract. Okay, so starting with the fed state. In the fed state, that's when you're going to be activating these segmental contractions, which makes sense because if you're eating, you're getting new stuff into your small intestine, and you're going to want to start breaking that stuff down so that you can absorb it. So that is going to be done by segmental contractions because it does the mixing, and the mixing is going to help with digestion. Whereas peristalsis is going to be involved with moving undigested material towards the colon. So they're going to be activated during different times. So associate the fed state with the segmental contractions that are done by the circular muscle, and is involved with mixing the chyme so that it can come in contact with all of the little enzymes that are also in here, and so that they can be broken down better compared to just, if they're all just sitting there. And once again, your segmental contractions are coordinated by your interstitial cells of Cajal, and it's the basal electrical rhythm or the slow waves, whatever you want to call it, that are going to be activating this. So you can change the BERs based off of either parasympathetic or sympathetic control. So parasympathetic is going to activate them. Sympathetic will likely inhibit them because it's your parasympathetic nervous system that's going to be responsible for digestion. So they can be controlled through those two autonomic nervous system mechanisms. But yeah, I think that's all for this slide. Okay, so now moving on to peristalsis. So peristalsis is that electrical activity is going to be known as your migrating myoelectric complex because it's the movement of your electrical activity down the intestine. So it's going to need to move things in farther, like it's coordinated so that the electrical activity moves in one direction versus mixing where you have like this way and then that way and then this way, and it's like more of like a localized area. So you should associate the term migrating myoelectrical complex with peristalsis. However, these are still being stimulated by your interstitial cells of Cajal, making these basal electrical rhythms, or your slow waves as they're also called. So associate those as the same thing, or just know that your interstitial cells of Cajal do it for both of them, peristalsis and segmentation. Okay, so after segmentation is done, that's going to occur after you finish absorbing everything and digesting everything. At this point, you want to take what wasn't digested or wasn't able to get absorbed and move it down towards the colon so you can get rid of it. So you don't have to memorize the 2 feet or that it takes 2 hours. I mean, you can generalize that it takes 2 hours, but Don't worry about memorizing those numbers, but what you should know is motilin. So motilin is going to be secreted by M cells, so that's really easy to remember, M and M. And that motilin is going to be activated during the fasted state. So that's another main point to take away from this. So whereas segmentation will occur in the fed state, peristalsis is going to occur in the fasted state. So that's like another difference between the two that you can like make a note of. But yeah, so your M cell is going to secrete motilin. It can also respond to a motilin agonist to secrete motilin, and that's going to stimulate peristalsis. So that can help move things along your intestines. So this is just kind of showing you what your migrating myoelectric complex looks like. And so it makes sense that you would have electrical activity firing in kind of like a wave-like manner. I don't know how to explain it, but you start firing a lot in the top part of the duodenum. So this is supposed to like represent the beginning of your duodenum. And then as it moves things along, it'll increase firing in the second part of your duodenum. So you'll see more firing there, and it'll continue to move things along until it works its way to the end of the jejunum. So that's just all that's saying. Don't worry too much about it. It's just really just showing you how your migrating myoelectric complex looks like. So I guess the only other thing to take away from this slide is knowing that cathartics can alter the amplitude of these, so they can cause your small intestinal muscles to contract harder, and that can act as like a laxative by increasing the amplitude and duration of these, so you can move things along better. So that's all. I don't think he'll test you on that, though. But you should definitely know this vocab word and associate it with peristalsis. And the fasted state, of course. Okay, so this is really just saying the same thing, where your M cells are going to be activated to release motilin, and motilin is going to stimulate peristalsis. But this is only going to be stimulated to be released under fasted conditions. And then after that, during fasted conditions, it's going to occur in a cyclical manner. So this is what's showing, it's just showing you like this cyclical manner of this. So every 80 to 120 minutes while you're still fasting, you're going to stimulate release of motilin, and that's going to move things along in like a cyclical manner. That's all I would take away from this slide, honestly. Okay, so this is a good summary slide, I think. You don't have to know all the details. I did highlight the important stuff, though. So your duodenum, we know, is going to secrete CCK because it has the I cells, it has the S cells as well. And it also has the GIP cells, but he also calls them the K cells. I don't remember which term he prefers, but I guess know both. And then also bicarbonate is going to be secreted here through the pancreas. So good summary slide here. You'll see most of your digestion occurring in your duodenum, and then it'll start to decrease as you work your way towards the ileum, which makes sense because all your food starts off here, so you're just going to have most of your digestion here. Same thing with absorption. Don't worry about iron absorption. What you should know is that your bile acids and your vitamin B12 are going to be absorbed in the ileum. And I think B12 is like specifically the terminal ileum, so that's an important point to know. Bile acids, just like generalize those. Your ileum is also going to be responsible for secreting peptide YY, which is important as well, as we'll learn later. And then you'll have motility of both segmentation and peristalsis throughout the entire small intestine, depending on whether you're in the fed state or in the fasted state. Okay. I guess that's, yeah.
I'm sorry, did you say there was a second name for K cells?
I think he also called it GIP cells, so it's really just named after what it releases.
Okay.
Yeah. Okay. Okay, so you guys already learned this in histo, so it should be really easy. So you have your villi, which are the bigger ones. And then you have your microvilli, which are on top of the villi, and they're the really tiny ones. So it's the microvilli that are going to make the biggest difference in increasing surface area. But I think he has another slide for that point later. But both of them together are going to increase your surface area, which is really important for absorption, since these are all made up of cells, and these cells have your transporter proteins to absorb material into the bloodstream. So if you're talking about, carbohydrates or amino acids or amino acids, they would enter the cell and then be absorbed by the bloodstream. So you can see that the bloodstream runs through here, but you also have your lacteal, and your lacteal is going to be responsible for absorbing fat, so free fatty acids in the form of a chylomicron. So yeah, just that's the only other point I would take away from this. But note that you have blood running through here and also lymphatic lacteals running through these as well that help with absorption. And then he mentioned that chyle is the milky substance that is inside of this, and that's because it's filled with chylomicrons, and chylomicrons are mostly composed of triglycerides. So it gives it that like milky appearance because it's mostly fat. But that's all I would take away from this slide as well, especially since you guys know this already. Okay, so you should associate celiac, Crohn's, and ulcerative colitis as being barrier function issues that can lead to leaky gut. And these are all caused by different things. I honestly don't remember the difference between celiac and Crohn's and ulcerative colitis, but you learn that in patho next summer or this summer, so don't worry about it. But they're all going to ultimately lead to the destruction of the tight junctions between the cells, and that allows for things to leak through the paracellular pathway. So you have your two cells, they have the tight junctions between them that keep them together, but if you destroy them, it allows things to flow between the cells. And that's not good because A, you're not absorbing things if they're leaking between the cells, and B, like that can cause an inflammatory response and can cause more destruction to your GI tract. So it's really bad. And this is known as leaky gut. So you should associate barrier function issues with the term leaky gut, and that it has something to do with destruction of the tight junctions and movement of things through the paracellular pathway. But yeah, that's all I would really learn about this as well, because he mentions, I think, Crohn's and ulcerative colitis later as examples of IBS. But yeah, okay. Don't memorize these numbers. Don't worry about that. I would even not memorize these numbers either. The point is he wants to make here is that your microvilli are going to be the ones that exponentially increase the surface area. So they're like the ones that mostly do, but he does want to like emphasize the fact that you have all of these things occurring to really increase the surface area to maximize absorption of nutrients. So he already kind of like introduced the aspect of having like a tube in like the first lecture and how that helps increase surface area. But then you've got your folds of Kerking, and then your other folds, your villi with your microvilli, and it just maximizes surface area. So that's all I would take away from this slide, especially since you guys already learned it in histo. Okay, so this is now getting to the more important stuff, and this is where I would probably focus like a good chunk of my studying, because you do have to know some of these transporters and the differences between absorption of the 3 different nutrients that he gives you. Okay, so you should know luminal absorption. So this is going to occur for proteins, it's going to occur for fat, and it's going to occur for carbohydrates as well. So all of these are going to have luminal digestion, which makes sense because your pancreas is going to dump out all of those enzymes, and you'll see lipases, amylases, proteases, and that's what it's talking about when it talks about luminal digestion, is the digestion that is occurring in the lumen. As the chyme is mixing the enzymes in with the chyme, and it's breaking everything down during that time. So during segmentation. The next type of digestion is brush border, and I think this is only specific for, I believe it is just proteins and carbs. So proteins have brush border digestion and carbohydrates do as well. Fats only have luminal digestion. And then the last type, so brush border digestion has a cell on the brush border that has a enzyme that is connected to the plasma membrane of that cell that can help break things apart. So protein, it can break it into a amino acid or a di or tripeptide. And then lastly, cytosolic. This is specific to protein only. So this is going to be breaking down di and tripeptides into amino acids through intracellular proteases. So your enterocytes do have intracellular enzymes within them that can break down di and tripeptides into amino acids. And remember that you cannot absorb anything bigger than amino acids into your blood. So you have to make sure that you break down proteins all the way down to amino acids before you can absorb them into your bloodstream. Okay, so this is just comparing and contrasting the three different ones. As we already discussed, proteins do all three of these because they have enzymes at the brush border. They have enzymes secreted by the pancreas, and they have intracellular enzymes as well that break them down. Carbohydrates can only do luminal and brush border, and then lipids are going to only do luminal because they have like a very special way of being broken down. So remember, lipids get broken down all the way down to fatty acids. And then as fatty acids, they can diffuse across the cell membrane because fatty acids are hydrophobic. So once they enter the cell, that is when they would enter the endoplasmic reticulum of that cell, get converted back into triglycerides. So it kind of seems counterproductive, but I guess the purpose is to break them down so that they can diffuse across. And then as triglycerides, they get put together in a chylomicron, because remember your chylomicron, when we learned about that in your LDL, HDL lecture of cholesterol and biochem, your chylomicron is made up of predominantly triglycerides, and that's how you transport fat throughout your body as a triglyceride. So it does get reformed in a triglyceride, put together as a chylomicron, and then it enters the lymphatics through that lacteal. So make sure you know that. That's super high yield information.
Phoebe, where does the micelles come in that again?
So I believe that's in the lumen. So fats are only broken down in the lumen, and they that's like you're breaking down the fat further and further. They become micelles, and then they get broken down into fatty acids and then diffuse across and then get put back together as triglycerides.
So they come in as lipids, they go down to triglycerides, then micelles, and then they kind of just go backwards?
Yeah, pretty much, except they don't make micelles again, I don't think.
Right, they make chylomicrons. Okay.
Yeah.
All right, sweet, thanks.
Yep. Okay, you should also know how these get absorbed into the cells. So proteins need to rely on active transport mechanisms, meaning they need ATP. And so when they enter the cell, they will go through transporters that require ATP as either dipeptides, tripeptides, or amino acids too. And then they have to get broken down into amino acids within the cell before they can enter the bloodstream. So this requires ATP. Carbohydrates cannot enter as anything bigger than a monomer. So it has to be glucose, it has to be galactose, it has to be one ring. That's it. Like it can't be like lactose or anything like that. So they have to be broken down all the way down to a monomer. So another thing to note is that glucose also gets taken up through active transport similar to proteins. However, fructose is taken up through facilitated diffusion, which is easy to remember because fructose starts with F. Facilitated diffusion starts with F, so you can remember it that way and just like generalize everything else as being active transport, except for lipids because they're hydrophobic, so they can just diffuse across. So that's what's mentioned here. So your lipids, your glycerol, and your free fatty acids will diffuse across the cell after they are broken down entirely in the lumen into these two, and then they just diffuse across, and then they get re-put back together as a chylomicron. Okay, so starting with carbohydrates. So in the lumen, you start off as some kind of starch. The amylase that's secreted in your saliva and your stomach and your pancreas ultimately breaks it down into oligosaccharides and disaccharides. So these at the brush border can further be broken down. So you have specific amylases that are connected to the brush border of your enterocytes. The examples he gives you are maltase, lactase, and sucrase. I probably wouldn't memorize these unless you have extra time to do so, but I don't think he would do that to you guys where he tests you on these. But just note that there's specific amylases, and so these would, and also disaccharidases and monosaccaridases, but sorry, not monosaccharide, but disaccharidases. But anyways, it breaks it down into monosaccharides, and now as a monosaccharide, that can enter the cell because it's small enough to do so. So you obviously don't have certain enzymes to break down certain types of bonds or carbohydrate type bonds. So certain fibers like plant fibers we can't break down, so that does not get broken down to a monosaccharide, and therefore we can't absorb it. So this is what is termed as fiber in our digestive tract. And then these are the simple sugars along with fructose that we can take up through active transport. Not fructose though. Fructose is facilitated transport. Okay. Yeah, and then this is just emphasizing that you can't take up disaccharides. They have to be monosaccharides. Okay, so we already have to know like a similar transporter here in the kidney. So I would probably memorize this transporter, and I would also memorize this one. And this one, don't worry too much about it because that's like, you know, taking it into the blood. So that's the lumen. This is the blood side. So as we are breaking down our glucose and galactose, we ultimately get them down to monosaccharides. So these two are going to use sodium as a co-transporter to enter. Remember, this is also active transport, so it does require ATP. And remember that the duodenum was responsible for getting everything to be isotonic because we do rely a lot on sodium to help help absorb things. So this is one example of that. So do know this transporter. Don't get it confused with the kidney one, which is number 2. This one's number 1. And then fructose, you can remember that as being GLUT5. It's easy because 5 starts with F. Fructose starts with F. So just anything that has an F in it, you can just associate it with fructose at this point. And don't worry about how fructose enters because they really don't know how that works. But anyways, everything that's an oligosaccharide that we couldn't break down because it was like plant fiber or whatever, that is going to continue down the intestines and ultimately will be excreted out because we can't break it down, and therefore we can't get it down to a monosaccharide, and therefore we can't absorb it. Okay, yeah, and then he mentioned something about SGLT2 inhibitors, which obviously would decrease glucose or galactose absorption. So make sure you know at least these 2 transporters, not so much this one. Okay, don't worry about these graphs, just kind of generalize everything. Main point here is that most glucose is absorbed in the duodenum, which kind of makes sense because we already talked about how glucose can get absorbed really quickly compared to like lipids and proteins. So obviously that's going to happen mostly in the duodenum, so we don't have a whole lot left to absorb by the time we get to the jejunum. So the reason why this is like a wider, like it's not even a line, it's kind of like this, like, I don't know. But it's just showing you like the differences in the population. So some people, like this is the minimum, this is the maximum. And so like the average would be somewhere like around like here. But yeah, for the most part, just generalize this graph as being that you absorb most of your glucose in the duodenum. Okay, so for lactose, it varies a lot because some people don't have the lactase enzyme, as you guys have probably learned about already, like so many times. You don't necessarily have to memorize the population of people who tend to not have the lactase enzyme. But he may use it in a question stem to kind of hint to the fact that he's talking about a lactase deficiency. So it might just be worth like looking over real quick. But yeah, for people who can absorb or break down lactose, you'll see that it's going to happen mostly in the jejunum. You wouldn't obviously absorb lactose, but you would see most of its breakdown is going to happen in the jejunum. It gets broken down into glucose and galactose. So that would mostly happen in the jejunum, and that's where you would see the absorption of the galactose and glucose that got broken down from the lactose. Okay, but yeah, main takeaway is that some people can't like break down lactose because they don't have the lactase enzyme. Okay, so moving on to protein digestion. So we have proteases that get secreted from the pancreas. And remember, the most important one that you do need to memorize is trypsin. It starts off as trypsinogen, and it gets activated mostly through enterokinase. So enterokinase is a brush border enzyme in the duodenum, which makes sense because you wouldn't want it to be found in the ileum or the jejunum because that's too downstream, and then you wouldn't have enough time for trypsin to like break things down. So obviously it's found in the duodenum where you see trypsin being excreted into through the pancreatic secretions. So enterokinase, do memorize this term, it's important, is going to activate trypsin, and then trypsin is going to be responsible for activating a bunch of other enzymes, lipases, other peptidases, that sort of thing. So once again, protein digestion, proteins get absorbed into the enterocyte, so between the lumen and the blood. They have to do this active transporter that gets absorbed into the cell as either free amino acids, di, or tripeptides. However, in order to enter the blood, they can only be free amino acids. So they need to be broken down further. So these di and tripeptides need to be broken down further through proteases inside of the cell. So make sure you know that because that's important. And that's just saying that cytosolic degradation of these two is what breaks them down to free amino acids so that they can enter the blood. Okay, moving on. So I think I memorized all of these. Obviously, trypsin and chymotrypsin are like the most important ones. But I think it's worth noting because I think he has a table in a few slides of all of these and what they break down. And I think he mentioned you have to memorize them. So yeah, memorize the table. Don't worry too much about this, but know that trypsin is then going to activate chymotrypsin elastase and these carboxypeptidases as well. So that's all I would really get from this. Okay, so remember that your protein digestion is going to occur in all three potential places, in the lumen, in the brush border, and the cytoplasm. So remember that the lumen, not only pepsin in the stomach breaks down proteins, this acid also breaks it down. So this is inside of the stomach because remember your chief cells and your parietal cells are going to secrete these two to help break down proteins. And so by the time they get into the intestinal lumen, in the duodenum, they're partially broken down, but there's still probably some intact proteins. So you need these other pancreatic enzymes to be secreted so that you can break them down into oligopeptides. These can be broken down by brush border. So these brush border enzymes can break them down into either amino acids or di and tripeptides, but they need to be oligopeptides before the brush border enzymes can do that. And so ultimately you can take in di, tri, and amino acids into the cell where they would ultimately, well, the amino acids can enter the blood. but these would have to be broken down even more so intracellularly. But we've said that so many times, so I'm sure you guys get it by now. Okay, so this is also important to know that taking up like di and tripeptides is dependent on proton. So you have this sodium proton antiporter similar to what you have in the kidney that is going to pump out protons, and these protons are going to be like co-transported back into the cell. So it's kind of like cycling around like that to help absorb di and tripeptides. So you should know that there is, you don't have to like memorize the name, but you should know that this antiporter is going to help with uptake of your di and tripeptides. And then those di and tripeptides will have to go through other peptidases to be broken down into amino acids to be absorbed. But yeah, main takeaway is that you rely on protons in order to take up di and tripeptides. Okay, and so most of your absorption of proteins is going to be in the jejunum as well. And then he also said that 1/3 of energy of food is used for digestion. So by eating protein, it's gonna take up so much energy to absorb it and break it down that like you only really like get a net 2/3 of energy from actually eating protein. So that's why it's kind of like good if you're on a diet to eat a lot of protein. But yeah, just generalize these graphs like I already said. Okay, so lastly is lipid digestion, and he has like 2 pictures to kind of show this better. So you could use either picture, but he's not going to test you like on the specifics of the picture. It's just to help you visualize it better. But you should know that once again, lipid digestion only happens in the luminal phase. So in the lumen, it's not going to have brush border enzymes, and it's not going to happen intracellularly. And it's going to rely on the bile salts from the gallbladder, your pancreatic lipase, colipase that also gets secreted, I think, from the pancreas, and then phospholipids, which all together break down large fat globules into smaller fat globules, and then ultimately into micelles, and then they ultimately get broken down even more into free fatty acids and a monoglyceride that would ultimately, diffuse across the cell membrane, and then be put back together as triglycerides into a chylomicron. So I already explained that. So these pictures should help you kind of put that together. So here's your bile salt. It's got some a little bit more polar material to it, so it helps break things apart and keep the fat globules like separated from each other so that they don't turn into a giant fat globule again. So it helps it break it apart into smaller droplets. So that is the action of these 2 that help take a big fat globule and break it into smaller fat globules. So that's what I would note as the actions for bile salts and phospholipids. So this is what happens after that. So I don't really like this picture particularly. You don't have to know like all of these like specifics. It's just kind of showing you like the steps for each of these. But ultimately you would break it down into, this is supposed to mean monoglyceride. So that's like the glycerol backbone of the triglycerides, and then your fatty acids. So these 2 can easily diffuse across, and then be reformed back together into a triglyceride. And these apolipoproteins, remember these are the proteins that go onto your LDL, your HDL, and your chylomicrons that kind of tag it and give it its identity. So that's all that's saying. So then you would just put those together into a chylomicron, and these would be absorbed in the lacteal. Super important to know that. I prefer this picture personally because it kind of puts everything all together instead of, breaking it up into 2 slides. So once again, you have a big fat droplet and you emulsify it with your bile salts and your phospholipids into smaller fat droplets. And then your bile salts and pancreatic lipase can, further break these down into micelles. This is all luminal because remember, it's all of this like digestion for fat is going to happen in the lumen, not at the brush border or anything like that. Ultimately, you'll break these down into your monoglycerides and your free fatty acids, which will then diffuse across, enter the endoplasmic reticulum, get put back together, repackaged as a chylomicron. And this chylomicron, which starts off inside of the cell, will exit the cell and enter your lacteal. So I don't know. I like this slide better than the last two slides. So I would probably use this one for studying this mechanism. Okay, and then most of your fat absorption will happen in the jejunum as well, which is where most of these occur. So your protein and your fat are mostly absorbed in the jejunum, and then glucose is mostly in the duodenum. Okay, so moving on to the water and electrolyte absorption and secretion in the small intestine. Most of that occurs in the large intestine, the colon, but a little bit does occur in the small intestine. So we already talked about how sodium is important for nutrient absorption. And the specific example we saw was for glucose through this transporter here. But it's also important in the proton antiporter. So sodium enters, the proton leaves, and that helps with di and tripeptide absorption again. So that's another example of how sodium is coupled to nutrient absorption. You'll also see that the uptake of sodium can also drag chloride with it through paracellular routes because of the charge difference. So, if you pump up sodium in one direction, this negative charge of chloride is going to want to follow it, and then water is going to follow both of them. So that's really just what I would take away from that. And then, yeah, just water is going to follow the solutes, as we've already seen so many times. Okay, so don't worry about memorizing these numbers. You can generalize this picture as well. When you're digesting, you will secrete water through gastric secretion, saliva. So you're losing water and putting it into your lumen. But at the same time, your small intestine and your colon are both responsible for reabsorbing a lot of this fluid that you put in there. So in general, you're not losing that much fluid daily through these secretions, even though you do secrete a lot because you do wind up reabsorbing it. That's the main takeaway from this slide. So don't worry too much about the specifics. Okay, so he does want you to understand this particular channel as well. So your cystic fibrosis transmembrane conductance regulator is going to be responsible for pumping chloride out into the lumen. And this can help drag sodium through paracellular pathways and water. So it's going to help move water from from our body out into the lumen. And so it's going to add water to the lumen to help move things along if it needs to. Another thing he mentioned here is that, what happens with cholera. So I don't know if he's actually going to ask you any questions on what happens with cholera, but basically the cholera toxin is going to upregulate this channel. So you wind up pumping out a bunch of chloride. And if you're pumping out a bunch of chloride into the lumen, we know water and sodium are going to follow. So that's why you'll get really bad secretory diarrhea from that. And you'll also lose a lot of electrolytes and water, and you can wind up being really dehydrated. So I think that was something he may have wanted you to know. And yeah, I think that's it for this slide. Okay, so super high yield, make sure you know this. I know there is a test question about it, and I vaguely, I don't remember if it was a practice test question that my TA gave us, or if it was actually a test question, but I vaguely remember somebody asking a question about like if you like resect somebody's terminal ileum, what would happen, and it had something to do with decreasing vitamin B12 uptake. So maybe keep that in the back of your mind as well. But anyways, so when you eat something that has vitamin 12, it enters your stomach and it's going to first bind to this R protein. He underlined haptocorin, so I would probably remember that term instead of R protein. So haptocorin is going to bind to your B12 and kind of protect it from the acidic conditions in the stomach. At the same time, remember your parietal cells are responsible for secreting intrinsic factor. So as your B12 is moving through your duodenum, it's still attached to this haptocorin. And then some point in your, I think, duodenum, it gets like released from it, and then that's when it's going to bind to your intrinsic factor. An intrinsic factor is then going to take it all the way down to the terminal ileum, and it's going to help it bind to the receptor so that it can be absorbed by the enterocyte. So it's super important that you know that these two, both your haptocorin and your intrinsic factor, are responsible for B12 uptake, and that if you don't have B12, that's going to impact your metabolism and your red blood cell production. So it can give you anemia. I think he may have used anemia as the example in the question stem, or maybe I'm thinking of another class. I don't know. We've learned this so many times. So yeah, just note that it's going to impact metabolism and cause anemia if you don't have B12. He didn't underline DNA synthesis, so I would say that's lower yield thing to memorize. Okay, so I added this as a flowchart because I thought the last one was kind of like chaotic, but it's pretty much saying the same thing. Okay, and then ulcers. He wants you to know the difference between a gastric and a duodenal ulcer. He also mentions H. pylori. H. pylori leads to the breakdown of your mucosal lining, and so it allows the stomach acid to reach your stomach lining and cause an ulcer, and that can be bad if it's severe enough to like go all the way through your stomach lining, so that you wind up having like gastric secretions being leaked out into your abdominal cavity, and that's very dangerous because it can destroy your other organs. But anyways, your gastric peptic ulcer is simply going to be an ulcer in your stomach. And then your duodenal peptic ulcer is going to be a ulcer in your duodenum. So he gave you a few causes for this. So this could be caused by too much hydrochloric acid and pepsinogen secretion in the stomach, which makes its way to the duodenum, and that can be too acidic for the duodenum. Can't secrete enough bicarb to neutralize it, and you wind up with an ulcer. Also increased parietal cell mass. So if you have a ton of parietal cells, more than usual, like, I don't know, under like cancer conditions, I don't know, that can also lead to increased hydrochloric acid secretion, and you wind up with more acid in your small intestine as well. Okay, so just know the difference between these two and have a general idea of the causes of this, and you'll be fine. Okay, so make sure you go through his summary. I also made notes of like the things he was talking about at the end of his lecture, so like the high yield points, so I just made notes of those for you guys. And I think that's it. Does anyone have any questions before I move on to the next lecture? Alrighty, if not, I will move on. So this one's shorter. It won't take us as long. So go through his learning objectives. I think he spends a lot of time on those to like point out the important stuff, so make sure you go through these. All right, so this is a review of what we learned in the last lecture. It gives you a little bit more information and connects previous topics as well, so I like this slide, so I would definitely look at, spend some time on it. But anyways, so your duodenum has both your I cells and your S cells. So your I cells are going to be the CCK secreting ones, and your S cells are going to be the ones that secrete secretin. So your S cells are going to be responsible for detecting high concentrations of protons, so low pH and acidity. And this is going to tell it to secrete secretin, which makes sense because you want to neutralize the acid, so it's going to respond to acid by secreting bicarb. And remember, bicarb, kind of like how it works in your salivary glands, bicarb is mostly secreted by your duct cells. So yeah, if you learn your salivary glands, you can kind of like generalize those with your exocrine glands in your pancreas because they secrete similar things by the similar cells. They're a little different, I guess, but you can kind of like learn them together. Okay, and then CCK is going to respond to fats and peptides, as we already discussed. So it doesn't respond to carbohydrates, just fats and peptides. And that's going to tell it to secrete digestive enzymes from the pancreas. And these are the acinar cells, because those are the ones that are typically secreting the enzymes. And it's the same kind of situation in your salivary glands as well. Okay. So remember when we learned about like our gastric and cephalic phases? These can also trigger pancreatic secretions. Remember, your cephalic phase can also trigger this. The only thing it doesn't do is it doesn't relax the sphincter of Oddi. So you wouldn't see bile secretion through the cephalic phase. But your gastric phase does that, and then it also will help with pancreatic secretions as well. So this is through the vagus nerve, also uses the DVC to help mediate all this. So together, they will lead to pancreatic secretions. So this is a good summary slide for like the past 3 lectures, I guess. Okay, so this is really high yield. All right, so if you eat something that has like carbs or very sugary, those are going to be entering the duodenum. So some glucose is going to be broken down through the amylases in the stomach and the saliva. So some of that is going to be broken down. Remember, most of your glucose will be absorbed in the duodenum. So you will see a lot of glucose within the duodenum. And in the duodenum, you have these GIP cells, also known as K cells. So you can remember both names. I think he added this, so maybe he prefers K cells. I don't know. But anyways, this will respond to glucose, and it's going to release GIP. It's easy to remember GIP. Don't get it confused with GRP, different things. But GIP has an I in it, meaning it's probably having to do with insulin. So I for insulin, and just associate it that way. So GIP will enter your bloodstream, and it's going to then go to the islets of Langerhans and activate beta cells to then secrete insulin into the blood. Glucose can also be directly absorbed in the duodenum, enter your bloodstream, and then it itself can go and activate insulin secretion from the B cells. So note that these 2 mechanisms together work to secrete insulin. Another thing he mentioned was that only oral glucose can do this, obviously, because once it's in the bloodstream, it's going to act through this mechanism here. So it's just going to go directly to the islets of Langerhans and lead to insulin secretion. It's not going to have any contact with the GIP cell. So I guess that's another thing he wants you guys to differentiate between. Okay, so now we're talking about the exocrine pancreas. Note that the last slide was talking about the endocrine pancreas, so don't get those confused. Okay, so pretty much this is kind of a low yield slide, so I wouldn't really worry too much about it. The point is that you aren't very good at secreting enzymes when you get older, so you can run into some digestion problems as a result of that with old age. And then he also mentioned that older people have a decrease in muscle mass, which may be due to the fact that they don't secrete as many pancreatic enzymes, so you can't break down proteins, and so you can't absorb as many proteins, so you wind up having a lower muscle mass due to that. So yeah, low yield, I would say. Okay, so this is just an anatomy review. I'm sure you guys already mastered all of this already. But it seems like he really wants you guys to know the different sphincters. Definitely know your upper and lower that we already talked about. He spends a lot of time on the lower esophageal sphincter, so I would probably, really get that down pat. So then you've got your stomach, you've got your antrum here. It's the lower part of your stomach, and it's like right before your pyloric sphincter. So your pyloric sphincter is what is opening up to, empty out your stomach into your duodenum. So although he doesn't really mention it specifically in any of his other slides, you should, know that the pyloric sphincter is really important for gastric emptying. And then you've got your sphincter of Oddi. This is only going to be opened up during the gastric phase specifically, and probably your intestinal phase as well, but not during your cephalic phase. So make sure you know that because he mentioned it several times as well. Your liver makes the bile, your gallbladder stores the bile. You will see gallbladder contraction both during your cephalic and your gastric phase. But, your sphincter of Oddi only opens during the gastric phase between the two of those. And then you have your small intestine, which then empties into the colon through the ileocecal valve, and then ascending, transverse, descending sigmoid, and then rectum. I wouldn't really worry too much about the internal and external anal sphincters because he does not talk about those at all. So I think he's just talking about like these sphincters that he, really focused on. Okay, so this is kind of an overview, just reminding you of the difference between your exocrine and your endocrine pancreas. Your exocrine is going to be responsible for secreting your enzymes and your bicarb. It's made up of your acinar cells, which are these, and then your duct cells, which are these. These are the ones that secrete bicarb. These are the ones that secrete enzymes. And then your endocrine cells are going to be accumulating together as the islets of Langerhans. You've got a mixture of alpha and beta cells here. Your beta are the ones that secrete insulin. Alpha are the ones that secrete glucagon, but he doesn't really talk about glucagon that much. But yeah, note that they're very similar to your salivary glands. So you should, it'll be easy to remember both of them that way. And then these are the enzymes that your pancreas will secrete. Okay. Okay, so this is the summary table that I guess I was talking about earlier. I think he wants you to know these, so make sure you know what each of these enzymes do and what they act on. So your trypsin, chymotrypsin, and elastase, you can clump these together. Obviously, they act on proteins because we already talked about them being proteases, and they're specifically going to break peptide bonds in any region of the proteins. Well, we know that trypsin and chymotrypsin have specific amino acids that they work on. So it's just generalize it as being just peptide bonds that they break. Your carboxypeptidase, they're going to also break off proteins, but they're going to break off the carboxyl end of a protein. So you have a protein that starts with the N and then goes all the way down to the C. So it's going to cleave here and it's going to wind up releasing the last amino acid in a chain of polypeptides. So that's the difference there. Lipase is going to break apart fat into your glycerol and fatty acids. So you get 1 glycerol and 3 fatty acids per triglyceride that you break apart through lipase. Amylase, now we're talking about your carbohydrates. So these are going to act on polysaccharides, specifically going to create glucose and maltose through cleavage. And then I would say these are lower yield to remember because these first 4 are really the ones that he focuses on. But these are really easy to remember, so you might as well remember them as well. Your ribonuclease and your deoxyribonuclease, so these are going to work on RNA. These are going to work on DNA. And you're going to get your nucleotides. Pretty straightforward. Make sure you know this table because he did want you to know it. Okay. So similar to the salivary glands, you have your acinar cells and your ductal cells in the exocrine pancreas. So your acinar cells are going to be responsible for secreting enzymes into the duct, and they can also move these other cations and anions across, but that's not as important. So they are very similar to salivary glands because you will see sodium and chloride and bicarb being excreted from your acinar cells in your salivary glands. But the main point he wants you to remember for the exocrine pancreas is that these are going to be the ones that secrete the enzymes, whereas the ductal cells are mostly responsible for secreting your bicarb. So yeah. Don't worry about all the other details in this picture. Okay, so this is, I don't think this is specifically an acinar cell because you see secretin acting on it, but I think it's just like a general cell from your like pancreas that it's just kind of generalizing everything and putting it together. But anyways, point is all of these can act on pancreatic cells to help either secrete bicarb or secrete enzymes. So we know secretin is going to be responsible for secreting bicarb, and all of these, I believe, are responsible for secreting enzymes. So it's really just secretin that is not really involved here. I think he just added it, honestly. But anyways, yeah, so VIP, we remember that as being associated with nitric oxide. It's going to be released, you know, through the DBC. It's like neuronal activation that releases VIP. GRP is also released at the end of a neuron, and so is acetylcholine. So these are all technically like neurotransmitters, I guess you can think of them as, and it's through, you know, your brain actually releases these to stimulate pancreatic secretion. And then these two, your CCK and secretin, as we already discussed, are going to be activated through the cells in the duodenum, your I cells and your S cells. So that's the difference. You don't really have to know which ones are CAMP mediated and which ones are calcium mediated, but you can remember that if it makes you feel more comfortable. I don't think I did, and I don't think he asked it. I don't think that would be very fair, but you can if you want, if you have extra time. Okay, so again with secretin, it's kind of like a negative feedback loop to help minimize, you know, acidity in your duodenum, because you don't want to like make it too acidic to potentially destroy the cells in the duodenum. So your stomach will release the contents into the duodenum, and with it, it's going to be mixed with acid. So once it enters your duodenum, it gets picked up by the S cells, and that tells the S cells to secrete secretin, which then enters the bloodstream and makes its way to the pancreatic ducts. They bind to their secretin receptor on the pancreas, and the pancreas is then stimulated to release bicarb to help neutralize the acidity. So he's said so many, like he's talked about secretin so many times, so I would say that this is definitely something that he's going to test you on. Okay, a little less important, but still something to keep in mind, is the action of mixing and its effect on triglyceride digestion. So your stomach does do some breakdown of triglycerides that you ingest, and it's going to be done through the actions of mixing that occurs in your stomach. So your stomach moving everything around and making sure to maximize the surface area so that your food comes in contact with lipases is going to help, you know, break apart your fats. So keep in mind that triglyceride digestion does depend on the stomach mixing it, and also on lipase, which is also released in the stomach. And then remember that it's going to be fats and proteins as well that can then trigger release of CCK, which then goes and tells your pancreatic enzymes to be released as well. So this is just telling you that if you can't mix your stomach contents, you'll likely not have as much triglyceride digestion, and then you'll have probably decreased CCK release as a result of that. Okay, a little more important is just re-emphasizing the role of enterokinase on activating trypsin. So your trypsinogen is your inactive enzyme, and so it's going to act on through enterokinase to be activated as trypsin, and then that's going to go and activate the rest of them. We've said this so many times, and He only mentioned that trypsin acts on this one, and these two in a previous slide, but now he's adding these two other ones. So really don't worry about memorizing this because there's some discrepancies here. But this is obviously going to happen in your proximal gut lumen because you want this, this is where you're secreting your pancreatic enzymes. And if you wait too long, you're not going to have as much trypsin activated. And then you'll have decreased breakdown of your protein. So you definitely want this to happen as soon as possible. Sorry, Lance, I didn't see your comment. Secretin acts on central acinar cells, which are inside the acinar cells and run along the ducts. So I guess acinar cell isn't completely wrong on that side. Oh, really? Is that too
Yeah, because that kind of like freaked me out too when I saw it. But yeah, I think he kind of combines them. But I remember in histo, I asked Veronowska about it. So it doesn't really matter for us for this class, but he does call them ductal cells instead of CNR cells that secrete and act on. So.
Oh, okay. Well, that's good to know because, yeah, he kind of like clumped everything together here.
Yeah, I just put that in there in case anyone can make that distinction from histo, which we just took last week.
Okay. Well, yeah, thanks.
Yep.
Okay. So moving on to the next slide. So this is just talking about how how CCK acts to activate gallbladder contraction and bile secretion. So CCK does a lot of things. This is when you're keeping a list of all the stuff CCK does. This is something to add to that list. So when the food that is high in lipids and protein come into contact with the CCK cells, it activates CCK, which then goes to through a vagal afferent. So it activates a vagal afferent to then go and tell the DVC, which is the brain region that we need to know for GI tract, to go and then not only contract the gallbladder, but also relax the sphincter of Oddi. So you should know which neurotransmitters are responsible for each of these actions. Remember, with acetylcholine, that's always going to cause contraction when we think of it in terms of GI. And then your VIP and nitric oxide together are going to cause relaxation. So this is going to squeeze the bile out, and then your VIP is going to let it enter the duodenum. So I added gastric phase here because this is just to let you know that the gastric phase can also lead to contraction of the gallbladder. So it's somewhat independent of CCK, but together they will lead to gallbladder contraction. Yeah, and that's it. And remember that your cephalic phase will not lead to relaxation of sphincter of Oddi. But I've said that like 100 times, so hopefully you guys already know that by now. Okay.
Hey, Phoebe, the cephalic phase, it does constrict the gallbladder a little bit, though, but you just don't get the sphincter of Oddi opening, right?
Yeah, exactly.
So both phases will
constrict. Yeah, I probably should have put cephalic as well. You're right.
Yeah, I just remember that. All right, cool. Thanks.
Mm-hmm. Okay. This is moving on to the other thing that CCK does, and this is not only does it act to release bile through the gallbladder, but it's going to then act on the pancreas through the same vagal afferent and efferent to release acetylcholine, VIP, and GRP. So these are the three that are released through this pathway at the pancreas to release enzymes. So this is specifically how CCK leads to the release of enzymes. So that's just like the specific mechanism of action. Okay, so make sure you know that. Know what activates the CCK cell, know where it acts through the DVC, and know what the DVC releases in order to stimulate enzyme secretion at the pancreas. And I think you should be fine. Okay, so moving on to the large intestine. So this is where he mentions the ileocecal sphincter. So this is going to be where the large intestine and the small intestine meet. So know what that is. I don't know how he could ask a question on it, but I don't know. Maybe he'll think of something. So this is going to be mostly involved with absorption of water and electrolytes. And then you also have like some kind of hormonal control of this as well. And I think the example he gives you is aldosterone on that. But anyways, by this point, your small intestine has absorbed as much as it possibly can. So this is going to lead to the leftover undigested material to enter your colon. And in your colon, you have all of this colonic bacteria. The colonic bacteria is super important because it's going to be able to break down this undigested material because they have the enzymes for it that we don't. And it's going to convert these into short chain fatty acids, super high yield point, because I think there was a question on the exam about short chain fatty acids. So what these short chain fatty acids do are then released from the bacteria, and they go and feed our colon cells. They're called colonocytes. I think the first time I learned this, I thought that colonocytes were also bacteria, but they're not. They're the cells in the colon. Okay, so your gut bacteria also does release neurotransmitters, which is why they're doing a lot of research on the, connection between your gut and your brain, because these can, alter your neurotransmitters in your body. And then they also can help make vitamins. But the main one you want to know is your short chain fatty acids. And then we'll later learn about the action of peptide YY, which is also something you definitely need to know. I don't know why he didn't underline it here, but it is very important. So I would probably know it for sure. Okay, so now we're talking about peptide YY, and I don't know why it's cut off like that. Whatever. So when your free fatty acids that have not been absorbed yet, so they're broken down, they can be absorbed, but they haven't been absorbed yet because I guess they haven't, come in contact with a cell on the lumen. But, if you have something that you eat that's high in fat, and it's going to take a lot longer for it to be broken down and absorbed. So if it makes its way down to the terminal ileum, it's going to come in contact with these cells here, and they're going to recognize the free fatty acids and release peptide YY. Peptide YY will then go back and slow everything down. So you're going to see less gastric acid secretion, and that's going to help decrease gastric emptying so we can stop moving things along, because if you just add more stuff in, it's going to move everything out, and so then you're not going to be able to absorb these extra free fatty acids that haven't gotten the opportunity to become absorbed. So yeah, like that, you also decrease pancreatic secretion, and together it's going to decrease your movement towards the colon. So the point of that is just to allow for more time for these free fatty acids to be absorbed before they're pushed along, and then they can't be absorbed. You don't have to memorize all of these steps, but just have a general understanding that it's going to slow everything down. So you can pretty much guess on the questions or the answer choices if, he gives you a question on something like this. Like if it's going to increase movement of your GI tract, then it's probably not the right answer.
Hey, Phoebe, real quick question on that. Just for specifically the gastric acid secretion, wouldn't you want to, I guess I just don't understand why it wouldn't increase, because I would think that would help with absorbing or I guess breaking down any of the food.
Yeah, so I guess the point is that since these free fatty acids are likely, it takes 2 hours for the meal to move all the way down here. So I think by that point, it just assumes that everything's been moved along. But yeah, your free fatty acids have already been broken down to their smallest, parts. So it's just an absorption issue, not necessarily a digestion issue. So I don't think that gastric acid secretion would necessarily help that very much. Because you don't really need to break it down anymore. You just need to give it more time to be absorbed. Does that make sense?
Got it. Yeah, I think that makes sense. Thank you.
You're welcome. Okay. So know your different parts of the colon. I'm sure you guys have this mastered by now, so I'm not going to spend much time on it. And then he did mention that the descending colon has the highest concentration of gut flora. So this is where a lot of your short chain fatty acids are going to be made. That's, what I would take away from that. Okay, so you do need to know the difference between caudad and orad, because I think that's, the technical terminology for the directions for these. But it makes sense because if you're pushing against the normal movement, you're technically heading back towards the mouth. So orad is that direction, and then caudad is like caudal, so towards the tail. So that would be the normal forward way direction. But anyways, you should know the differences between the different regions of your colon. So you should know that the ascending colon is going to have most of the absorption of the water and electrolytes. And it does so by pushing everything down towards the orad direction. So make sure you associate orad with ascending. And by doing so, it's going to kind of squish everything towards the bottom. So it kind of wrings all of the water out. Okay. Sorry, Savannah, let me just read your message. It slow down to increase the time the bolus comes in contact with digestive enzymes. That makes sense. Increase time and increase surface area, maximize absorption. It's like a feedback. Yeah, that's exactly correct. Thank you for sharing that, because I may not have explained it very well. But yeah, if anyone's still lost on that, that's a perfect explanation. Okay. So then your transverse and your descending colon are going to be involved with moving things in the caudad direction, so towards the rectum. So yeah, just know the difference for the ascending and then the rest, and you should be fine, and associate the ascending with being able to maximize absorption by pushing everything down and kind of like wringing it out, I guess you can think of it as. Okay. So you should know your 3 different types of movements or contraction patterns. So when food first enters the colon, it's going to still be mixing to try to absorb as much water and electrolytes as possible. So mixing is going to allow us to do that. And so this is going to have no real direction. It's kind of doing like segmentation movements, but this is in the colon, not in the small intestine. And then your haustral movement, this is where you're going to see your ascending really starting to do that orad movement to help wring out as much water as possible. And then you'll also continue to see a little bit of mixing, and you'll see towards the end of your descending that you're going to see movement towards the rectum. And then your mass movements is the last one. And this is going to be triggered by a large meal. Your gastrocolonic reflex is associated with this mass movement because remember, your gastrocolonic reflex is going to want to make more room. After you eat a large meal, it's going to trigger movement of the colon to want to make you poop so that you wind up making more room for the food that you just ingested. So it's going to promote this mass movement, which is going to really push everything towards the descending colon and promote defecation. So at this point, because you did most of your absorption in your ascending, you'll see that the contents are a lot drier. So you have to have stronger contractions in the transverse and your descending to help move things along, because as you can imagine, if something's drier and denser, it's harder to push it along. So that's another thing he mentioned, and something that I would just generalize as well. So make sure you know the differences between these 3 and then the order in which they occur. So this is 1, 2, 3. Okay. And then he also mentioned, before I skip on to the next slide, that these can be dysregulated in someone with IBS. So IBS is kind of an umbrella term for every kind of GI issue. So, if you have something that causes a neuronal issue that causes you to not be able to do these, it can, be a potential cause of IBS. Okay. So a little bit more important is understanding, how everything moves along. So like your entire GI tract, so as we talked about in the stomach and the small intestine and all of that. These have your interstitial cells of Cajal. These are the pacemaker cells. They're found pretty much everywhere, and they're going to be the ones responsible for setting the pace of these muscular contractions. So once again, they're the ones generating the slow waves, or your basal electrical rhythm. Same word, same thing, but different words for it. When you have increase in sympathetic stimulation, it's actually going to downregulate the firing of your interstitial cells of Cajal. And as a result of that, you're going to see less action potentials and reduced muscle contraction in your colon. So you won't be moving things as much during sympathetic stimulation, which is pretty straightforward, I think. Okay, and then this is supposed to represent like normal conditions where your parasympathetic is the one mostly taking charge. So this is parasympathetic, this is sympathetic. Okay, so last vocab word that you do need to know is your gastrocolonic reflex. So this is when you eat a meal. Remember, you have those stretch receptors on your stomach. You also have stretch receptors and other cells, you know, in your duodenum, like your CCK and all of that, that can be activated and tell your CNS that you just ate something. So then that can lead to neuronal efferents that can alter the activation of the muscle in your colon and promote motility within it. So ultimately, it's going to promote movement of everything down and out. And the purpose of this is to make more room for this meal that you just ate. So yeah, and then the other thing that I would take away from this is that not only can you stimulate it through neural afferents, but you can stimulate it through hormones as well. So it can be activated in 2 different ways. He doesn't say which hormones do it, but so don't worry about that. But yeah, you're going to just see both neural and hormonal control of the gastrocolonic reflex with the purpose of moving everything forward and out so that you can make more room for your new meal.
Phoebe, on this slide, is the afferents going to the DVG, or is it just from like a stretch receptor, like a mechanoreceptor in the stomach? I don't know if
yeah. So I think it is a stretch receptor. I don't know if he like actually said so, but I'm assuming it has to be a stretch receptor. But you can also have the hormones, and also CCK can also tell the DVG that it's going. I'm assuming it's talking about the DVC because that's the one that really we focus on. But I don't know if he actually like emphasized what part of the brain it goes to.
I wrote DVG. I don't know if that's from him saying that or if I looked that up. I can't remember, but I have DVG.
Okay, yeah, then it must be DVG because that's the one that he really focused on for all of the GI tracts. So when in doubt, just guess DVG.
All right, thanks.
Yeah. Okay, so aldosterone. He mentioned this briefly. I don't know if he actually will test on this, but just in case. So aldosterone works kind of the same way in your colon as it does in your kidney. So if you remembered it for the kidney, you'll be fine for the colon as well. So remember, aldosterone is going to stimulate sodium reabsorption, and wherever you have sodium reabsorption, you'll also see water reabsorption as well. So this is just a way of absorbing more water in the colon. So people who have hyperaldosteronism, who are secreting too much aldosterone, will have increased sodium reabsorption and therefore increased water reabsorption. So they're more prone to constipation because of this, because they reabsorb way too much water, and so you can't move things down the colon as easily without it. So that's just, I don't know, something to know, I guess. Okay. So once again, talking about your short chain fatty acids. So here's your luminal side. Here is the blood side. So remember, your gut bacteria are going to take undigested fiber and then convert it into short chain fatty acids. And these short chain fatty acids are also going to be co-absorbed with sodium. So remember, sodium is super important for absorbing a lot of our nutrients. And so they're very important for short chain fatty acids. So these short-chain fatty acids are going to be metabolized in the colonocyte to help make AATP. And so that continues this, gradient where you're moving sodium out because sodium's moving down its gradient here. You're moving sodium out, so you're going to move more sodium in, which brings short-chain fatty acids with it. So essentially, short-chain fatty acids will kind of upregulate themselves through, making ATP to continue this, gradient effect. So you should definitely know this transporter, the SMCT1. I feel like that's important, and he underlined it, so it must be very important. So make sure you know it, and you can, and know that it, works with sodium reabsorption. The only thing he really wants you to know for short-chain fatty acids is that they fuel the colonocytes. And then there's more research going into their other effects, which are all of these and how they can be like tumor suppressors and all of that. And then he also mentioned that probiotics and prebiotics can help, make more of these bacteria and promote better gut health. And these bacteria can make more short-chain fatty acids. That's another effect that probiotics have. But main point is knowing that these are going to give fuel for the colonocytes to make ATP and to work properly. Okay, so obviously don't memorize the Bristol stool chart. That's too much information, but you can kind of generalize all of these. I would say that these are the more high yield points, because if he does ask a question on constipation, it'll likely, include like the information in this first 3 bullet points is kind of like part of the question stem or something that you have to like infer. But I don't think he would ask something about constipation. I feel like that would be somewhat low yield, but these are pretty obvious. So obviously low fiber can lead to constipation. A high calcium concentration can also, like if you eat a lot of calcium, which is also associated with dairy, which can both cause constipation, and then dehydration can also cause constipation. Keep in mind that it is calcium that's going to, calcium in acids, not like magnesium or anything like that, that's going to cause this constipation. And then opiates, we all kind of know that opiates are going to typically lead to constipation. So people who are on long-term pain medication with opiate pain medication, or people who do heroin can struggle with constipation as well. As for these diseases, I really wouldn't worry about memorizing these. I just highlighted Hirschsprung's because we've learned that so many times. And then IBS, definitely know it because he does have, a couple slides on that as well. Okay, so moving on to diarrhea, he definitely wants you to know the 4 types of diarrhea. And then you can kind of generalize the causes. So a lot of the causes for diarrhea are viral, bacterial, or parasitic. And most of a lot of children in the world die from that because they don't get good water. And so they are prone to having parasitic or bacterial infections that can cause diarrhea and then dehydration and then death. So he does mention that inflammatory bowel disease, or IBS, IBD, whatever you want to call it, or Crohn's, can also lead to diarrhea. So this is through an autoimmune disease. So destruction of your cells in your intestines through this can lead to malabsorption. So you can't absorb things as well. And as a result of that, you're going to have diarrhea. Food intolerances, so the example he gave is lactose intolerance, because you don't have the enzyme to digest this, the lactose, so that can also cause diarrhea. And then ischemic bowel disease, so if you're not getting blood flow to a part of your colon, that decreases the surface area, so you wind up with malabsorption. And that can also lead to diarrhea, and then colon cancer can also do it too. So definitely know your 4 types of diarrhea. So your secretory diarrhea is going to be some kind of issue with your colon, or with your colon. So either you're pumping out too many solutes, such as in the cholera toxin, upregulating your CFTR channel and increasing chloride excretion, or there's some kind of other issue that is causing you to secrete too much water into the lumen. So osmotic diarrhea is a little bit different. This isn't going to be caused by an issue with your intestines, it's likely due to you taking something that doesn't get absorbed and it draws water with it, kind of in the same way that like glucosuria causes increased urination. So same kind of concept here. A good example of this is like prune juice. So prune juice has sorbitol in it, and sorbitol doesn't get digested, so it just draws a lot of water out with it when it's going through the lumen. So that would cause osmotic diarrhea. And then motility-related diarrhea is usually due to some neuropathy or some kind of like nerve issue that causes you to not move things as well. And if you can't move things, you can't digest, and then you wind up with malabsorption. And then inflammatory, so this is due to like food poisoning, due like some kind of bacterial or parasitic infection or viral infection, something like that, where it's inflammatory cause. So know the differences between the four. I would say this is low yield, definitely low yield. But these are like different treatments for diarrhea. So obviously if you have like anti-motility agents, you can't move things along, then that would stop diarrhea because you can't move anything. And then you have Pepto-Bismol, which is used for many different things. It's used for heartburn because it acts as a buffer, and then also has anti-inflammatory effects. So that's how it could treat like inflammatory diarrhea, for example. And then he also mentioned this about traveler's diarrhea, and he talked about his story of how he takes antibiotics before traveling, but don't worry about that. And don't worry about all of this stuff. Maybe note that this is more like kidney related, that if you do have diarrhea, it can lead to metabolic acidosis. So that's just something to know mostly for the kidney aspect. But everything else I would say is pretty low yield. Okay, so IBS. IBS is an inflammatory condition, and it's kind of an umbrella term for a lot of other inflammatory conditions, which includes Crohn's disease and UC, which are like really common forms of irritable bowel syndrome, like the ones that we all know. So this is due to some autoimmune response, and that autoimmune response ultimately leads to damage to the intestinal wall. And if you damage those cells, you have less surface area to absorb things in. And so that leads to malabsorption. Anytime you have malabsorption, it's likely going to lead to diarrhea. So here he just mentions this like just once, but I feel like it was important. So your bile acids, we already kind of talked about how they get reabsorbed in the terminal ileum. So these bile acids are going to influence electrolyte transport, which you didn't really talk about how that works, but just associate bile acids with electrolyte transport, and how if you can't reabsorb these, they're going to leave your body and they're going to draw a lot of water out with them. So that is why, not being able to absorb bile acids can lead to diarrhea. Okay, don't memorize this. So with IBS, it can be associated with either constipation or diarrhea, and these are all different potential things that can be associated with IBS. So they all kind of contradict each other. So if you have like increased slow waves, that's going to increase motility, but then decreased myoelectric response to feeding could decrease motility. So it's like kind of contradicting itself. So don't worry about memorizing these. Just kind of associate IBS with causing issues with many different things that can either speed everything up and lead to malabsorption, or slow everything down and lead to constipation. But yeah, low yield. Okay, make sure you go through the summary questions because I think that's super important because he goes over like the important stuff. Okay, so any questions before I go through the last Kahoot?
I'm still kind of confused now. I'm like where is bile? I know where bile like kind of goes, but where is it doing like its main action in the GI tract?
So bile is mostly going to be breaking apart the lipids, which I guess mostly happens in the jejunum. So it's going to break apart the lipids, and then some of it is going to make its way out towards the colon, and then it would be reabsorbed in the terminal ileum, I believe. So you can like recycle them for next time.
And it enters the GI through, doesn't it enter through the duodenum? Is that the sphincter of Oddi it also goes through?
Yeah, so bile is being excreted from the gallbladder.
Yeah.
yeah, it would enter the duodenum. You will see some breakdown of fat with the help of bile acids in the duodenum, but I think according to his like graph, most of it happens in the jejunum for whatever reason.
Okay, so it enters upstream, but it doesn't really start doing its function until midway through the small intestine.
Yeah, for the most part, I would probably think of it that way.
All right, cool. All right, thanks.
You're welcome. All right, I guess I'll start the Kahoot if nobody has any other questions. Oh wait, I don't think I put the volume on it. Oh, that's nice, guys. I'll give you guys like maybe 1 more minute to join. All right, I think I will start it now because it looks like everybody's already in. Awesome. Yeah, Motilin is going to move everything down, so that's not going to slow gastric emptying. Good job. Awesome. Yeah. Okay. Remember, capillaries, you don't see absorption of fats through capillaries. Instead, you see it through the lacteals. Nice. Okay. Yeah. Remember, carbohydrates do both. Amino acids or proteins do all three, and then fat is only doing the lumen. Awesome. Yeah, fructose, facilitated transport. Okay, so it's going to be galactose. Remember, you want to make sure it's a monosaccharide, so galactose and amino acids can both enter the bloodstream. Fatty acids will not because they enter the lymph as a chylomicron, and then disaccharides are also too big, so they have to be broken down into monosaccharides like galactose, glucose, or fructose. So that is why those other answers are wrong. Awesome, yes. Just a fact to note is your ascending colon where you see most of the water and electrolytes because of that squishing action. Right, secretin is associated with bicarbonate secretion, and it's going to act on your ductal cells because that's where most of your bicarbonate comes out. Okay, yeah, so VIP actually leads to the dilation of it so that it opens, and then CCK is going to signal the release of VIP. So both of those are involved. Cephalic phase does not do that. It's a gastric phase that does that, and then GIP isn't related because it's associated with insulin secretion. Okay, yeah, so shifting movement is not one. Good job, guys. Okay, so hypothyroid can cause constipation. It's the magnesium and acid that would not cause constipation. So it's calcium and acid that does. Magnesium would actually do the opposite. Okay, so I guess this is goodbye unless I see you guys at my office hours. So good luck with the rest of the program, and good luck with the rest of your careers and everything. So feel free to reach out to me if you ever need anything or anything like that. But yeah, I'll stick around if anyone has any questions. And thank you for whoever put "We Heart You," Phoebe. Love the shout out, and congrats on winning as well.
Hey, Phoebe.
Yeah.
One of the questions you had talked about CCK and I think was VIP that relax the sphincter of Oddi.
hmm
Could you walk through what VIP is again? It's slipping my mind for some reason.
So VIP is really just something that leads to like he clumps it together with nitric oxide, and he just kind of uses it as something that dilates things. So it does the opposite of contraction. So he used it as an example for relaxing the stomach and relaxes the sphincter of Oddi. I think that was another example. And also during swallowing, I think it's also involved with like relaxing the esophagus to promote movement downward, if that makes sense. So just kind of clump it in your head as similar to nitric oxide.
All right, thank you so much. I appreciate that, and thank you again for everything. I'm sure I'll see you in your Q&A sessions.
Alrighty. You're welcome, guys. All right, if there's no more questions, I guess I'll go ahead and end the meeting. Feel free to show up to my office hours or email me if you have any questions prior to the test. And bye, guys.